All right, so let's talk about blood. Blood and functions of the cardiovascular system. So we've got, oh goodness, blood vessels, arteries and veins and capillaries, and the heart. So basically the job of blood is to transport and distribute oxygen, carbon dioxide. And basically that happens by way of circulating blood cells. By way of delivering oxygenated blood, all right, so let's keep going. So basically, blood is distributed through blood vessels, and the heart is what pumps that. Basically, serves as the pump for those vessels to carry blood and to move blood through those vessels. You got an overview here of the cardiovascular system, right? So you got the heart there pumping blood out through blood vessels all the way to every tissue, through every artery and vein, and then all the way back to the heart. So basically, blood functions in a number of ways. It is a fluid. It's a connective tissue, but it transports oxygen, carbon dioxide, nutrients, hormones, wastes. It's just very fancy. But it also helps regulate pH and ion composition by absorbing and neutralizing acids. So acids in the body, there is a component of blood, a buffering system, basically, that allows for neutralizing acids and maintaining a normal pH. My puppy's there in the background scratching. But also blood vessels can restrict fluid loss, and there's a cell in blood called platelets that helps with blood clotting. And then white blood cells defend against pathogens and toxins. So we're going to talk about 5 different types of white blood cells. And then blood circulates to particular areas by changing the size of blood vessels. Blood can flow to or from an area of the body to stabilize body temperature. Again, it's a fluid connective tissue, and the components of whole blood are plasma and formed elements. And in the whole body, you have about 5 liters, give or take, of blood. All right, so basically this blood, again, distributes carbon dioxide. I think I flipped ahead a couple of slides. Bear with me. I've got puppies asking for a lot of help today, so this may be a little bit disjointed of a lecture. So we talked about transport. It's a fluid connective tissue, and there's plasma as the fluid component and formed elements, the red blood cells, white blood cells, platelets. Blood is very viscous, so it's thick, and it's slightly alkaline, meaning a little bit basic. So the pH is a little bit on the basic side. All right, so keep going. The fluid, okay, there we go. I'm gonna move forward there. Again, it's a little disjointed. I've got 3 puppies to watch after today, so bear with me, guys. The plasma is the fluid component of blood. It forms about 55% of whole blood volume. So if you stick a sample of blood in a tube, more than half of that would be plasma. And that plasma is what transports kind of organic and inorganic elements like plasma proteins. For example, a plasma protein like albumin is a major carrier protein in the blood. And that carrier protein carries substances that can't circulate in blood on their own. So something that's like basically lipophilic or hydrophobic, something that can't circulate in water easily, in that plasma, albumin can carry it. And then there's other things in plasma, 92% of which is water. But then there's electrolytes like sodium and potassium. Sodium is Na, right? K is potassium. And then you've got calcium and ions there that are really important to the function of the body. The formed elements account for about 45%, but there's a range depending on and there's variability from human to human and from between males and females that accounts for whole blood. And hematocrit is a measurement of the percentage that these formed elements take up in whole blood. So it's basically the percentage of whole blood volume that is formed elements. And the formed elements are composed of 99.9% of which are red blood cells, and then less than 0.1%, excuse me, are white blood cells and platelets. And we're going to talk about the major functions of some white blood cells today, and then other white blood cells that we'll come back and talk about in the lymphatic system. So red blood cells, this 99.9% of the formed elements are red blood cells, and they have a really large surface area to volume ratio, and that allows them to exchange oxygen and carbon dioxide really efficiently. And oxygen is bound to hemoglobin inside red blood cells. So you can find hemoglobin within red blood cells, and that's basically the major transport molecule for oxygen. And red blood cells can kind of form stacks, like they line up in a row, so that through narrow blood vessels, they can kind of run single file through the blood vessel without jamming. And they're a little bit flexible, so they can squeeze through really small capillaries. So here's a stack of red blood cells. It's called a rouleau. That stack of red blood cells through a capillary makes it really easy for those blood cells to get through the capillary, but also they line up single file so that oxygen and carbon dioxide exchange is really efficient. And there's a hemoglobin molecule. So there's 4 subunits, 1, 2, 3, and 4. In that subunit, in each subunit, there's a heme molecule that holds an iron, and those molecules are really important to binding oxygen. So one hemoglobin molecule can bind 4 oxygens. If you know your blood type, you're a step ahead. So basically, blood typing depends on antigens that should or should not trigger an immune response. So antigens are cell surface proteins. So they're embedded in the cell membrane, and they act as a beacon. So they're on the surface of the cell. They're an antigen. They are a cell surface marker that the immune system, cells of the immune system, can recognize as self, meaning that they're supposed to be there, or non-self, they're not supposed to be there. And if they're self, the immune system ignores them. So there's antigens on the surface of your red blood cells that basically determine your blood type, and the immune system ignores those antigens. There's other antigens we'll talk about later in the lymphatic system that are not ignored by the immune system that your body attacks. The cells of your immune system attack them. The most important regarding blood type are an A antigen, a B antigen, and an Rh antigen. It's also called a D antigen. And basically the plasma of the fluid component of blood can contain antibodies that attack antigens on foreign red blood cells. So if you were an A blood type and somebody gave you B type blood, your body would produce B antibodies to attack that foreign antigen. And there's an example of what type of antigen would be on the surface of a red blood cell and what antibodies would be located in that plasma. And in saying that, if you give somebody the wrong blood type, what happens is a cross reaction or a transfusion reaction. That transfusion reaction is basically a result of giving somebody the wrong blood type. So not good. And the response is what's called agglutination, or clumping together. So agglutination, if cells agglutinate, they clump together. And that's really bad in the body because cells aren't supposed to do that. And when they do that, that can cause them to rupture. It's called hemolysis. Think hemo is blood and lysis is to break something. So hemolysis or hemolysis is the breaking or rupture of blood cells. And think about it. If you need blood and somebody gives you blood, but it happens to be the wrong blood type, and then hemolysis occurs because of agglutination, you could be in really big trouble. And that's called a cross reaction or a transfusion reaction. And this is what happens, right? So an antibody that's produced to a foreign antigen, so here's the B antigen, for example, basically binds to the antigen, and that makes those cells sticky, and they stick together, they clump together, they agglutinate, and then that triggers those, basically targets those cells for hemolysis or hemolysis. To complete blood type, we have the positive and negative. So if you know your blood type and you're A positive or A negative or AB positive or O negative, that positive negative is whether or not the presence of what's called the Rh antigen is on the surface of your red blood cells. So it's an antigen like the A antigen, the B antigen, but the positive negative is what we use to designate whether that's present, positive, or not, negative. So full blood type presents that positive and negative sign. So A positive and O negative. A positive would be positive for the presence of both the A antigen and the Rh antigen. O negative means there's no antigens on the surface of their red blood cells. So there's three possible antigens, A, B, and Rh. And if you don't have any of those, if you don't have A or B, then you have O blood type, meaning there's neither A nor B antigens on the surface of your red blood cells. And then if you're negative, O negative has no antigens on the surface of the red blood cells. There's a table that kind of illustrates percentages of each blood type. So in the US and other kind of territories. Blood typing. If you don't know your blood type, here's how you measure it, or how you find it. Basically, you mix a couple of drops of your own blood with an antibody solution. And if you get clumping, agglutination, that means those antibodies are binding to the antigens on the surface of the red blood cells, and you're positive for that blood type. So here's what happens. There's one blood type. So this is A positive. You have now 3 samples of blood. One sample here, one sample here, and one sample here. And in each well, you put also an antibody. So here's anti-A antibody, anti-B antibody, and anti-Rh antibody. Remember, antibodies bind to the antigens to cause agglutination. So if you see clumping, this is clumping right here, agglutination, then you're positive for that blood type or that antigen. So this is positive for A, negative for B, no clumping, and positive for Rh, clumping. So this is an A positive blood type. Take a look here. Clumping, 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 all agglutination. So this is AB positive. And look here, no clumping, no clumping, no clumping, O negative. Make sense? So anti-A and anti-B antibodies are basically present at birth. So if you ever give B blood or A blood to the wrong person, those antibodies are already ready to attack. So you have those already. Anti-Rh antibodies are only formed if an Rh negative person, somebody without the Rh antigen, is exposed to Rh positive blood. So weird, right? You have A and B antibodies circulating and present at birth, but Rh antibodies are only formed if your immune system is exposed to those Rh antigens when you don't already have them. So you'll mount an immune response. White blood cells. There are 5 types of white blood cells, but they share some properties among the five types. So in the bloodstream, they'll circulate only for a short period of time and then migrate into connective tissues. They are activated in the bloodstream, so they can remain inactive for a period of time, and they're only activated in the bloodstream. They are attracted to certain chemical stimuli. So there's what's called positive chemotaxis, and that's basically white blood cells will migrate towards a chemical that they're attracted to. And that can guide white blood cells to pathogens and help them destroy those pathogens or damage tissue and clear it or heal it where needed. And some of them are phagocytic, so they can engulf pathogens or parts of a pathogen. And phagocytes are 3 types: neutrophils, we're going to talk about these here shortly, eosinophils, and monocytes. Those are phagocytic white blood cells. And here's the 5 types: neutrophils, eosinophils, basophils, monocytes, and lymphocytes. So there's 5 types of white blood cells. We're going to talk about some of them specifically, like the granular leukocytes. The granular, meaning they have granules in their cytoplasm. And when you stain those cells, the granules absorb stain in a different way. So neutrophils are the most numerous of leukocytes, and a leukocyte is a broad term for a white blood cell. So there's 5 leukocytes. Neutrophils is a specific type of one type of leukocyte. Eosinophils are pretty rare, and basophils are even more rare. So remember, less than 0.1% of formed elements are white blood cells. So neutrophils are the most numerous, but basophils, look at that, less than 1% of just white blood cells, which is less than 0.1% of the formed elements. It's crazy, right? It's very rare. And take a look. So the granules stain kind of light pale here. They stain like a reddish, pinkish orange color in eosinophils. And then basophils have really dark stained granules, so dark that it almost masks the nucleus. And this is what the nucleus is here, right? All right, I gotta go take care of a puppy. See where it picks me up. There we go. Good. Okay, puppies are all taken care of. So let's keep working. The agranular leukocytes are monocytes and lymphocytes. And we're going to talk about lymphocytes a lot in the next chapter, in chapter 13. So if they have zero, if any, cytoplasmic granules, so they don't absorb stain very much. And monocytes are huge. They're the biggest white blood cell with a big kidney bean-shaped nucleus. And then lymphocytes are about the same size as a red blood cell, and have a big nucleus. So there's just a little bit of cytoplasm that you can see there. And they are basically providing cell-specific immunity. So they defend against an actual specifically recognized antigen. Some of the other leukocytes, like I'm going to flip back a little bit, neutrophils. Neutrophils are your first line of defense against bacteria, and they're phagocytic. So they can engulf a pathogen like a bacteria and then send signals to other cells to come to the site of infection and fight the infection. Eosinophils are also phagocytic, but they will respond to different types of pathogens. They're involved in the inflammatory response in allergies, and they can defend against things like parasites. Then basophils are a pretty big white blood cell, but they release this chemical called histamine, and histamine is basically responsible for the allergic response. So when you get like watery, itchy eyes when you're allergic to something, basophils are partly responsible for that. Monocytes differentiate into, they're phagocytic cells. So they can phagocytose a pathogen, and they can actually become, so monocytes recognize an antigen and then can become, basically mature into a macrophage. And a macrophage is what is phagocytic about these cells. And they can engulf pathogen or cellular debris that needs to be gotten rid of. And then lymphocytes we'll talk heavily about in chapter 13. They have some pretty big jobs. There's two different types of lymphocytes, T cells and B cells. The stimulation of red blood cell production is called erythropoiesis. So a erythrocyte is a red blood cell. The hormone responsible for stimulating red blood cell production is called erythropoietin. And the stimulus for the production of erythropoietin is hypoxia, or low oxygen levels. And that can be in response to things like anemia, low red blood cell or low iron content. Remember, iron is what helps bind to oxygen. Lack of blood flow to the kidneys, and the kidneys are is the site of erythropoietin production. A decreased oxygen content in the air, so low oxygen, and damage to the respiratory surfaces of the lungs. So if you're not absorbing or diffusing enough oxygen into the blood, you could have low oxygen content. And basically, red blood cells are produced in the red bone marrow. So erythropoietin, or EPO, stimulates red blood cell production. It's produced from the kidney, but then circulates to the red bone marrow to produce more red blood cells so that more oxygen can be carried. Platelets are the last type of formed element that are just cell fragments, really, but they're produced basically by clipping off pieces of a cell called a megakaryocyte. And there's little flattened discs, and they function in blood clotting. In this clotting response, basically I gotta go check on a puppy. All right, puppy crisis averted. So let's keep talking here. Maybe it's averted. So the process of basically stopping blood loss through a damaged blood vessel is called hemostasis. So if a blood vessel is damaged and cut, that blood vessel can be repaired and blood loss can be basically inhibited or stopped by way of hemostasis. And there's three phases: the vascular phase, platelet phase, and coagulation phase. Those three phases basically are responsible. Let me see what's on my next slide here. Yeah. The three phases of hemostasis, basically the vascular phase happens in the blood vessel, and the blood vessel basically constricts to prevent blood loss. And then platelets come in and form a plug in the damaged vessel. It's called a platelet plug. And they clot to form this plug and cut off blood loss from the blood vessel. And then the coagulation phase is basically the phase where the damaged blood vessel gets repaired. So those three phases are really important. A couple of problems that can happen in the blood. One's called an embolism. So basically blood clots are a good thing, but they can also cause problems. So blood clots may form in the bloodstream where even no injury has occurred. And basically a drifting blood clot, not one that's not stuck in a blood vessel, is called an embolus. And that might get lodged in a small blood vessel, and that would block off the blood vessel. Think about it. A small blood vessel needs to supply blood to a tissue. And an embolism is a blood clot that has blocked off a blood vessel, so that the tissue beyond that blockage is no longer receiving blood flow. That tissue damage in response to that is called an infarction or an infarct. And that can happen in the brain. And an infarction in the brain is called a stroke. And an infarction in the heart is called a myocardial infarction, which is a heart attack. So a blood clot that gets stuck in a blood vessel can cause tissue damage called infarction. And that in the brain, that's called a stroke. And in the heart, it's called a myocardial. Think myo is muscle and cardio is heart. So a myocardial infarction is tissue damage in the heart, which is basically a heart attack. A blood clot that attaches to the vessel wall where there's no damage is called a thrombus. And oftentimes that forms when platelets basically get attracted to a plaque. And remember, plaques can build up because of too much fat in your diet. So blood vessels can be occluded by fatty buildup on the inside of the blood vessel, and then it makes it easier for a floating clot to get stuck. And with regard to blood, iron deficiency anemia. So if you've heard of the word anemia before, you think maybe iron levels or red blood cell levels. But basically insufficient iron intake can cause iron deficiency anemia, meaning red blood cells are small and they transport less oxygen. If they're too small and they're not transporting oxygen, basically iron is responsible for binding to oxygen on hemoglobin. And if you don't have enough iron, then you can't transport oxygen efficiently, which means you'll end up hypoxic, and that can be a lead to very bad consequences. Pernicious anemia is basically a vitamin B12 deficiency. So B vitamins are really important, and you have to, basically that prevents normal stem cell division and prevents normal red blood cell production. And basically what happens is red blood cells end up abnormally large but oddly shaped, meaning they can get stuck in blood vessels a little bit easier than they normally would. And some clotting disorders can be caused by insufficient calcium and insufficient vitamin K. So a couple of blood disorders there, all related to something related in your diet and your nutritional intake. All right, so that finishes the first part of blood, the blood chapter. Sorry about all the puppy disturbances. It looks like they're down to sleep now. So hopefully that won't be the case in the next chapter or in the next part of the chapter, where I will see you soon.